Look at these 4 limbs: a human arm, a bat wing, a whale flipper, and a cat's front leg. They look completely different on the outside and do totally different things. But underneath the skin, they share the same basic bone structure. 1 upper bone, 2 lower bones, a cluster of wrist bones, and 5 digits. Why would such different animals share this pattern? Welcome to Sciesmic. I'm Matt, and today we're exploring comparative anatomy, how studying body structures reveals evolutionary relationships. When different organisms share similar anatomical features, it suggests they inherited those features from a common ancestor. By comparing structures in modern organisms and fossils, scientists can trace evolutionary relationships just like detectives following clues. Let's decode these anatomical patterns. Homologous structures are body parts that share a similar basic structure and embryonic origin, but may serve different functions. They're one of the strongest pieces of evidence for evolution. The vertebrate forelimb is the classic example. Whether it's used for flying, swimming, running, or grasping, the basic pattern is the same. Humerus, 1 upper arm bone, radius and ulna, 2 forearm bones, carpals, your wrist bones, metacarpals, hand bones, and phalanges, your finger bones. Your arm has this pattern. You use it for grasping, typing, throwing, and it's really versatile at doing things like manipulation. A bat's wing has the same bones, but the finger bones are extremely elongated to support a wing membrane. Same structure, different proportions, different function. Flying instead of grasping. A whale's flipper? Yeah, you heard me right. Same bones again, but shortened and modified for swimming. The flipper contains the same humerus, radius, ulna, carpals, metacarpals, and phalanges as your arm. And a cat's front leg uses this pattern for running and pouncing. Same bones as your arm and the whale, just arranged for quadrupedal locomotion. Here's the key question. If these limbs were designed independently for their specific functions, why would they all use the same bone pattern? Why wouldn't whales have a completely different flipper structure optimized only for swimming? The answer? They inherited this pattern from a common ancestor. The pentadactyl limb pattern tells us an evolutionary story. Scientists propose that all land vertebrates, amphibians, reptiles, birds, and mammals inherited the basic 5-fingered limb structure from a common ancestor that lived about 375 million years ago. Fossils support this. Tiktaalik, a 375 million year old fossil, shows the transition from fish fins to tetrapod limb. It has fin rays like a fish, but also bones corresponding to the humerus, radius, and ulna, found in all land vertebrates. It's a transitional form showing how the pentadactyl limb actually evolved. Now, over millions of years, natural selection modified this ancestral limb pattern for different purposes. Some lineages elongated certain bones for flight, like the bat. Others shortened them for swimming, like the whale. Still, others strengthened them for running, like maybe you or even a cat. But the underlying pattern remained because all these species inherited it from that common ancestor. Now, we've talked a lot about limbs, but it's not just limbs. Vertebrate skulls share similar bone patterns too. All vertebrates have a vertebral column made of repeating vertebrae. These anatomical similarities across different species reveal their common evolutionary heritage. Homologous structures often develop similarly in embryos, even though adult forms might differ. All vertebrate embryos develop pharyngeal pouches, for example. In fish, these become gills. In humans, parts become ears and throat structures. Similar embryonic structures suggest shared ancestry. Vestigial structures are reduced or functionless anatomical features that were useful in an organism's past and with their ancestors. They're like evolutionary leftovers or evidence that organisms have changed over time. Humans have a tailbone, the coccyx. That's all that remains of the tail our distant ancestors had. It's a fusion of 3 to 5 small vertebrae with no external tail extending from it. Why would we have tailbones if we never had tails? Well, we inherited this structure from ancestors that did have tails. Your appendix is another vestigial structure. In herbivorous mammals, the appendix helps digest tough plant material. In humans, it's small and serves minimal function, suggesting our ancestors relied more on plant material than what we do now. Whales have tiny hip and leg bones buried deep inside their bodies, completely disconnected from their spines and invisible from the outside. These remnant bones reveal that whale ancestors walked walked on land with functional legs. Whales walked on land. As whales evolved, though, to become fully aquatic, these bones became vestigial. Some snakes have tiny remnants of hip and leg bones, evidence that their ancestors actually had legs too. Flightless birds like ostriches have wings but can't fly, vestigial structures from flying ancestors. Vestigial structures make sense through evolution. Organisms inherit features from ancestors, but when those features are no longer useful, they may reduce in size or lose function, but don't disappear entirely. They wouldn't make sense if each organism was independently designed for its current lifestyle. Now, not all similar structures indicate evolutionary relationships. Analogous structures look similar and serve similar functions, but have different internal anatomy and evolutionary origins. They evolved independently through convergent evolution. Bird wings and insect wings both enable flight, but they're built completely differently. Bird wings are modified forelimbs with bones, muscles, and feathers. Insect wings are thin extensions of the exoskeleton with no bones at all. They evolved flight independently, so their wings aren't homologous. Dolphins, which are mammals, and sharks, which are fish, both have streamlined bodies with dorsal fins, but dolphin skeletons reveal they're mammals with lungs and a modified mammal bone structure. Sharks have cartilage skeletons and gills. Their similar shapes evolved independently due to similar environments. We call this convergent evolution. So here's the key distinction. Homologous structures have similar anatomy and embryonic development, indicating some sort of shared ancestry, even if the functions differ. Analogous structures have similar functions, but different anatomy and origins, indicating independent evolution, not common ancestry. Cactus spines, which are just modified leaves, and tree thorns, which are modified branches, both protect plants, but evolved independently. Again, analogous, not homologous. To determine evolutionary relationships, we focus on homologous structures, similar anatomy, similar embryonic development, similar position in the body. These indicate common ancestry, regardless of whether current functions differ. Comparing fossil anatomy to modern organisms reveals spectacular evolutionary transitions. Whale evolution provides a perfect example. Pakicetus, a 50-million-year-old fossil, shows a mammal with 4 functional legs, hooved feet, and a long skull, but its ear bones are uniquely shaped like modern whale ear bones, homologous structures connecting it to whales. Ambulocetus from 49 million years ago shows a more aquatic lifestyle. It has webbed feet, a stronger tail, and a longer snout. Still has functional legs, but more adapted for swimming. Dorodon, 40 million years ago, looks more like a modern whale. A streamlined body, tiny vestigial hind legs. The legs are too small for walking, but reveal that evolutionary history. Modern whales have only small vestigial hip and leg bones buried deep inside. The evolutionary transition from land to fully aquatic is documented in progressive anatomical changes through these fossils. Archaeopteryx shows the dinosaur to bird transition. It has feathers and wings like birds, homologous to modern bird wings, but also teeth, a bony tail, and clawed fingers like dinosaurs. Its anatomy bridges those two groups. Horse evolution shows progressive limb modification. Early horses had multiple toes. Modern horses have one enlarged middle toe, we call it the hoof, with small vestigial side toe bones hidden inside its leg. Fossil anatomy documents the transition too. Today, scientists can confirm anatomical relationships using DNA evidence. Remarkably, DNA comparisons match the evolutionary relationships suggested by comparative anatomy. Organisms with more similar anatomy generally have more similar DNA sequences. Humans and chimps share similar skeletal structures and about 98% of their DNA. Humans and whales share the pentadactyl limb pattern, and about 85% of their DNA is the same. The molecular evidence confirms what anatomy suggested. Scientists have discovered that homologous structures are often controlled by the same genes. The genes that direct limb development in mice are nearly identical to those directing limb development in chickens, bats, and even humans. Shared anatomy reflects shared genetic instructions inherited from common ancestors. Modern evolutionary trees combine anatomical data with molecular data. When both types of evidence point to the same relationship, we can be very, very confident about evolutionary history. New technologies like CT scanning and AI let scientists examine internal anatomy without destroying fossils. This reveals hidden similarities like the inner ear bones connecting different mammal groups that further clarify evolutionary relationships. Evolutionary developmental biology, or EvoDevo, shows that organisms sharing homologous structures often develop those structures through similar embryonic processes, controlled by similar genes. This triple match, adult anatomy, embryonic development, and genes, provides overwhelming evidence for common ancestry. Comparative anatomy reveals evolution's signature throughout the living world. Homologous structures, like the pentadactyl limb, show that diverse organisms share underlying anatomical patterns inherited from common ancestors. Vestigial structures provide evidence that organisms have changed over time, retaining reduced versions of features that were once functional in their ancestors. We distinguish these from analogous structures, which look similar due to similar functions, but reveal independent evolution, not common ancestry. Comparing fossil anatomy with modern organisms documents evolutionary transitions. We can literally see in fossils how whales evolved from land mammals, how birds evolved from dinosaurs, and how horses modified their feet over millions of years. The beautiful thing is that anatomical evidence, fossil evidence, and molecular evidence all tell the same story. When three completely different types of evidence all point to the same evolutionary relationships, that's some powerful scientific support. Now, who knows? Maybe you'll become a comparative anatomist studying animal structures, a paleontologist discovering transitional fossils, or an evolutionary biologist understanding how organisms are related. Now, don't forget to subscribe for more amazing science videos, and let us know down in the comments what anatomical similarity surprises you most. Thanks for exploring evolutionary relationships with Sciesmic. Want to explore more about evolution and biological relationships? Check out our complete science curriculum at sciesmic.com, where every student can learn, grow, and achieve.